Now that we have done the experiment for human DNA extraction and banana DNA extraction, we will move on to GST gene sequence. So on the left is the human GST gene. It's called glutathione S transferase alpha 1 or GSTA1. This sequence shown here is the mRNA version from the NCBI database and is the first transcript variant of the gene. And on the right, we can see that we have the banana GST gene, specifically from Musca acuminata, which is the Cavendish banana. It's labeled as GSTF1 or probable glutathione S transferase. This performs a similar function even though it comes from a plant. So, even though these two sequences look very different, they actually code for enzymes that do almost the same job in both species. Later on, when we run the multiple sequence alignment, we'll be able to see which part of the sequence are conserved, meaning they stay quite similar through evolution because they're important for the enzyme function.
We perform pairwise sequence alignment using a bioinformatics software tool called Emboss Needle. These are the 3 pages of its results. Using the embossed needle, we have obtained that the sequence identity of these 2 genes are 35%. The similarity percentage are also 35%. Its gaps are 54.1%. And the mismatches can be recognized. From the results, this is an example that shows the yellow circles as the gaps. It indicates that one sequence has missing bases which are represented by dashes while the other continues. Gaps can occur due to insertions or deletions during evolution or alignment optimization. Next, the green circle represents the matches. The bases will align identically in both sequences. In the alignment, these are indicated by a vertical bar, meaning the same nucleotide or residue is present in both sequences. Lastly, the red circle as the mismatches. The bases or amino acids at mismatched positions are different between the 2 sequences. In the middle line of the alignment, this position has a space or dot but not a vertical bar, which shows that they don't match exactly.
Hello, so now I will present about MSA task. So I use comparative genomics to analyze glutathione S transferase across diverse life forms such as human, Arabidopsis, rice, and banana. So GST actually are detoxification enzyme. So to compare them, I use MEFFT, a bioinformatic tool, to perform multiple sequence alignment. So in the slide, I highlight the conserved region in red box. So in the red box, it shows near perfect sequence identity. So it actually show highly conserved region that correspond to the G site, the active site that binds the molecule glutathione. Conversely, the region around the G site show high sequence variability. So this variable region is the H site, which bind to diverse toxic substrate. The G site is conserved because it is under intense profiling selection. So since GS H is the same in human and plants, any mutation here will cause a loss of function and the gene will be eliminated. So this actually reflects a functional constraint at the genomic levels. In conclusion, by applying comparative genomic method, we successfully demonstrate that the GST gene family follow an evolutionary model defined by a highly conserved core for universal function and for organism-specific adaptation.